Okay, so today's lecture, and also we're going to do some breakout groups as well today, is going to be focusing on introducing spectroscopy in the electromagnetic spectrum, and specifically focusing on UV-vis spectroscopy, or UV-vis absorption spectroscopy. So let's see if my pen's working. So our goals today is to have an understanding of just spectroscopy in general, which is the science and the measurements dealing with light matter interactions. And specifically related to this, since it's light, we want to understand the different regions of the electromagnetic spectrum and how they relate to what we measure in biological samples from a molecular level and imaging side of things. So how do those different regions interact with biological samples? We're also going to talk about the different components for spectroscopic instrumentation, so things like the sources, the sample holders, wavelength selectors, and detectors. And we're going to talk specifically about things that go from the UV to the IR, the different types of instrumentation that's used for that. And then we'll get into some details about UV-vis absorption and how it's used to measure concentrations of proteins, nucleic acids, different molecules. So starting with our very general instrument sketch for every lecture here, our source is going to be a light source. I'm going to use like h-nu for light sometimes, since that's the energy of a photon. Our sample here, we're going to talk about the sample holders, but we're going to talk about all the different molecules, molecular biophysics. So we're at the amino acid or the nucleic acids or the lipid level. And sometimes UV-vis is also used for cellular measurements, but with scattering. And we'll talk about that in detail. And then in terms of the detector, since we're using light and seeing how light is interacting with our sample, our detector is going to be detecting photons as well. And we want to convert those to electrons, so then we can get a readout with analog to digital conversion to be able to measure that on a computer. So then we have our readout here. So we'll be going over those in some details. So getting into spectroscopy, the fact that we're working with light, you guys all should have had E&M classes where light here being the electromagnetic spectrum, we're having an oscillating electromagnetic field, where we can see that we quantify the energies or these different regions based on the wavelength. So we can characterize it by the wavelength from the peak to peak with that oscillating field, but also which has units of meters. You can also characterize the energy of different regions of the electromagnetic spectrum based on frequency, which then has units of hertz. Also, you can use wave numbers, which is 1 over the wavelength. Typically, this is used in IR spectroscopy and reported as inverse centimeters. And then also reporting it as electron volts. So all of these different units can be used to describe the different ranges of the electromagnetic spectrum. But depending on the fields that you're in, the type of spectroscopy that you're using, people will use different units for that. So just be aware of that and the relevant equations for shifting between these. If you want to go from wavelength to energy, it would be Planck's constant over the wavelength, or Planck's constant times the frequency. Wave number is 1 over the wavelength. Frequency, speed of light over the wavelength. And also if you want to do the period, 1 over the frequency. For Planck's constant, you guys need a refresher, 6.626 times 10 to negative 34 joule seconds. The speed of light, 2.998 times 10 to the 8th meters per second. So all these different ranges of electromagnetic spectrum are used for different types of spectroscopy and different types of imaging. You guys should have seen these in different classes, and to do a recap, and we want to go over what these energy ranges are. How do they interact with biological molecules? So what atomic or molecular quantum transitions? So this would be things like, do they interact with a nucleus with electrons, intershell electrons, rotations, vibrations, and the like? And also, I want you guys to think about different imaging applications that these different regions are used for. And we're going to get more into imaging applications later in the semester, but I'd like you guys to discuss those anyway. So I have breakout groups. These are you guys' net IDs. I pre-assigned them. Hopefully you guys should go to the right breakout rooms. But I'd like you guys to discuss this for the specific regions of the electromagnetic spectrum. And then on the next slide, we're going to fill out this table out. So also pick one person from your group that will type out your results or your discussion in the chat box. So I'll break you guys into these groups. Take some time to introduce yourselves since we're not like in the actual physical classroom. And I guess I'll give you guys about 10 minutes to discuss this. Take some time to, before, I know you guys are all on your laptops, before jumping into Wikipedia or Google, first try to discuss this, use your brain before having the computer answer it, but you can use that after discussion for a little bit. If you guys have some extra time, feel free to discuss the other regions of the electromagnetic spectrum, and I'll call you guys all back in 10 minutes or so. And I'll be in the main room if you have questions. You can chat me, I can go into the break room, stuff like that. I haven't used this that much. Let's see if I can break you guys out. Any questions before going in the breakout rooms? Oh, come on. Okay. I guess the pre-assignment did not work. Let me try this again. Guess I'll assign you guys manually, so I'm sorry. It'll take some time, or randomly, if I can do that. I'm gonna open, guys. I think you guys can all join. I think Todd, Jack, and Andrew, I have to add you guys somehow. I guess Rose, Andrew, Todd, and Jack, if you guys can just stay out here, I'm going to try to figure, you guys can start your discussion out here. I'll mute myself and I won't pay attention, and I'll try to figure out these breakout rooms. So.
Wait, I feel like when it's asking for the.
Sorry.
Yeah, that works for me.
Okay.
So we're doing, oh wait, introductions first. So hi everyone, I'm Andrew. I'm a senior engineering physics, and yeah, that's me. Nice to meet you guys.
Hey guys, I'm Jack. I'm a rising fourth year, and I'm a nutrition major. Microwaves can penetrate into materials and deposit the energy below the surface. I mean, yeah, that's kind of already, we already knew that.
Oh wait. And tell me if wrong, but like oh wait, fuck, people are back. Damn it.
I'm just back because I clicked the wrong button. I was still supposed to be in my room.
Oh, okay.
Development of new UV-vis microwave assisted.
Oh no. Professor, can we go last by any chance?
Well, you are last, but that's fine. I mean,
Okay.
if you guys struggled with some of this, I mean, hopefully it's emphasizing that these regions aren't all well described. And you may have thought these in terms of, since I heard some of the microwave discussion, like you think about these in terms of electronics, but in terms of biophysics, you may not be as familiar, or concepts that you've thought about in chemicals, how do they relate to biophysics? So if you guys didn't have a clear answer, that's totally fine. But let's start filling out this table. And so whoever was the person for the chat, let's see, start with group 1 with the gamma rays. If you want to chat, or if you want to unmute yourself, that will work as well. But what energy range did you guys define for the gamma rays? What units?
So what we found was, well, for gamma rays, it was around like 100,000 electron volts, close to around that range. And X-rays would have been closer to, on the lower side, like 100 electron volts.
That's what you guys found, which is a good middle value. So I'm going to list these. I'm guessing a lot of you guys did electron volts. I'm also going to put, for everyone's reference, in wavelengths as well. And this is like the range that I found. So you guys are right in the middle of those. So these are definitely short. Because you guys had both X-rays and gamma rays, right?
Yep.
Yeah.
Okay. Okay, and then for the quantum transitions, for the how it interacts with matter, what do you guys find?
So that one we had a little bit more difficulty with.
Mhm.
I think we were unclear in this case what the photons were necessarily interacting with.
Yeah, so I was thinking in terms of if you have any type of sample, how does it interact with the molecules that are there or the atoms that are there? So this is the highest energy region of the electromagnetic spectrum. So it probes very high energy, I guess, aspects of molecules. So with the gamma rays, they interact with the nucleus, and X-rays interact with the inner electrons. So X-rays are so strong that they ionize atoms, they ionize molecules, where they kick off those inner shell electrons that are, you typically think are pretty stable and near the nucleus. And then what about the imaging? What do you guys find?
We kind of didn't really get to that yet.
Okay. So I'll say for gamma rays, they're actually used, if you think about, they interact with the nucleus, they're actually used with radionuclide imaging for medical imaging. So if you think about, I guess for like gastrointestinal imaging, that you might have to take like a cesium radioactive radio tracer. That's how gamma rays are used. And then also X-rays, I think, is pretty typical that, I mean, there's not even a good word for it other than X-ray imaging, but you have to think about how that has damaging ionization for those samples. Okay. So what about for the group that did the UV?
For the energy, so for the wavelengths, we had 10 nanometers to 400 nanometers. So that corresponded to 3 electron volts to 124 electron volts.
Mm-hmm.
For the quantum transitions, I know like the hydrogen atom is the electron transitions for that are on the order of 10 electron volts or less. So I guess outer electron transitions would be able to probe.
Yep. Outer electrons, or you'll also hear them referred to as bonding electrons. Yep, so electronic transitions.
And then for imaging, since it's down to 10 nanometers, I guess it would be anything less than that. So, and all the way up to 400 nanometers. So I guess a lot of just small molecules in general, we'd be able to image. Anything less than, well, I guess depends what wavelength you're using, but less than 400 to 10 nanometers.
Yeah, so I would say that's definitely true if you were thinking about what its potential is for imaging things. But I'm also going to draw a line through this because UV is so damaging to biological samples, it's typically not used. So I guess a trick question in the UV spectrum. So if you think about any DNA mutations and the like, that's all caused by UV. And I also want to emphasize that people typically divide the UV up into far, mid, and near. So this terminology of near means it's near the visible region. You can see the same is done in the IR. So to break out those ranges, the far is 190 to 280 nanometers, and then 280 to 315 is the mid, and then the near is 315 to about, I guess, 400 nanometers is how that's broken up. Okay. Then what about the visible range group 3?
Yeah, so for energy, we got, it's of course 380 nanometers to 740, you know, going from purple to red, I guess, in visible light. And in terms of quantum transitions, I guess I only briefly looked at like the hydrogen atom and the sort of Balmer series electrons. So
Also, it's also
okay,
electronic transitions like the UV.
right. Imaging, we talked about bright field imaging and then optical microscopy, which, yeah, just goes down to I guess the max resolution is about 1/4 of a micron.
Yep. So the visible region, I guess, is the easiest one since that's what we interact with all the time, but that's why it's in most microscopic techniques. But yeah, you can see here, even like with the UV, we say it ends at 400 and 380, that these edges of the electromagnetic spectrum, depending who you talk to, that will vary. Yeah, that's good. Okay, so let's move on to the infrared with group 4.
Hi. So for IR energy, we said about 5 eV to 1.5 eV. That's near IR. I think if you're going out to far IR, it can go up to like e to the -3, something like that.
Yeah, I would say, yeah, that's more, what I'm looking at. My notes that it can even go down to like milli eV, I would say. So I'll say in my notes, I put in terms of wavelength around 780 nanometers to 1 millimeter. So yeah, this is more of the near IR. Then what about the quantum transitions?
The quantum transitions are probing the specific vibrational states of molecular bonds in biomolecules.
Vibrations? And then what about applications in imaging?
FTIR, so chemical analysis.
And I think with infrared imaging, I think an important point is that it's used for tissue imaging, or it's tried to be used because there's a tissue transparent window. And this is in, I think, the near IR, I think around 1200 nanometers, that light can pass through tissue. So you can actually get pretty deep imaging in the infrared region. So that's why that's used. So again, there's like a breakout of these different ranges of the infrared. I want to bring specifically highlight the terahertz region, since that might not be a region you've heard of before, emphasized. And that goes from 10 microns to 1 millimeter. In this region of the IR, longer wavelengths, lower energies, it doesn't have much interaction with matter, and there's not really great optics for this. So this is I guess a newer field of spectroscopy and imaging. I'd say, don't take my word for it, maybe like 20 or 30 years, where they're really trying to find detectors and optics that work with this field. Is a challenge, but interesting area of research. Okay. And then let's go to group 5 for the microwaves and radio waves.
All right, I think, so for the energy ranges we got for microwave, it was 1.24 millielectron volts to 1.24 micro electron volts, I believe, if anyone has. And then for radio waves, we got 1.24 micro electron volts to 12.4 femto electron volts.
It's the lowest energy there is. So wavelengths, this is about 1 millimeter to 1 meter, and then 1 meter to like kilometers. Okay. And then what about quantum transitions?
So like at first impression, we were really confused because we were thinking that like when we imagine radio waves, we imagine like astronomy, like probably like very large distances. But through some research, we found some interesting research that's been done with some sort of microwave and radio frequency spectroscopy, and we found a website that said that oh no, oh wait, yeah, okay. So using microwave transition frequency, they looked at geometric structures of molecules, electric dipole moments, energy barriers, internal rotation, centrifugal distortion parameters, magnetic moments, and just other information that they could find, and also information related to electron distribution and bonding. So that's kind of what we found for the quantum transitions. So it's possible
for
that
microwaves,
even...
all I could find. Oh, sorry. Continue, Todd.
No, go ahead. Yeah, go ahead.
Oh, for microwaves, what I could find were maybe bond rotation, as well as I read weakly bound complexes.
Yep, so rotations typically with microwaves. So you guys want to think about how your microwaves heat up your food. It's causing the water molecules to rotate, and that's how it heats it up. And then radio waves, very low energy, but they're applied to looking at spins of electrons for nuclei that are aligned in a magnetic field. So that's pretty specialized. We're going to get into the NMR discussion later on, but that's how radio waves are used for spectroscopy.
Oh yeah, also, oh yeah, I think my entry also sent me something saying that they also look at rotational transition for molecules in the gas phase. So I guess, I don't know if that's,
yeah.
So, yeah.
So think about, rotations is how microwaves. And then in interest of time, there is microwave imaging. Again, that's going to be limited by the wavelength that you have here. So that's going to look at larger structures than optical microscopy. So this is on millimeter range. And then for the radio waves with these spins and the nuclei, that's used in MRI imaging. So again, longer scales there. So table is pretty filled out. Do you guys have any questions with this? And I think always keeping in mind higher energy, smaller wavelengths, and then thinking about your energy diagrams for atoms and molecules. These are going from the highest energy to the lowest energy. Okay. So with these different regions of the electromagnetic spectrum, you'll use different components and different sources within the optical instruments to work with these different wavelengths and energies, and it'll depend what type of spectroscopy you're doing. So these components that I'm going to go over mostly are things that are used from the UV to the IR. And your source in these instruments obviously has to produce light. And in the UV to the IR, you can use lamps, which are continuous. And there's lamps like a tungsten lamp where you're heating up a tungsten filament, and that's going to have black body radiation. You can also think of xenon or other halogen lamps where you pass a current through there and it emits light from those electronic transitions. There's also deuterium or hydrogen lamps that again, passing current through there will excite it, it'll emit light, and these will work in different ranges of the electromagnetic spectrum. You can also use lasers, which, if you guys didn't know, laser is a abbreviation for light amplification by stimulated emission of radiation. And this is going to be a discrete light source. So that means that it's going to have a very specific single wavelength. It's going to be coherent as well and collimated as it exits. So this is a diode laser that I show here versus the lamp. And lasers can be made based on semiconductors with diodes. They can be made from gases, from dyes. But I would say like diode is the most common now. Over the past 40, 50 years, the cost by using semiconductors and making diode laser brings the cost way down. And then even, I have here in these little drawings that a flame can act as a source where you pass through certain elements, and by burning them, those elements will emit their own photons there. So these are common sources. If you're using a range of wavelengths, you'll more typically see lamps. If you're just trying to use one specific wavelength, you'll see lasers used more. But if you are using lamps, you have to be able to select the wavelength that you're using to pass through your sample, or you can also use this after your sample. And that will depend on the type of spectroscopy you're doing. So the cheapest wavelength selector would be just a very simple filter. These are glasses filled with dyes. or plastics. And they're a way to cut off and absorb some wavelengths of light and allow others to pass through. More complex would be an interference filter, where these are layers of glass, metals, dielectric, then another layer of metal and glass. With these different refractive indices of these different layers, certain wavelengths will constructively interfere, while the other wavelengths will destructively interfere. So that's how those work. And then monochromators. These would be your filter, your prisms, or your diffraction gratings that again take advantage of constructive interference with light. And I would say these are the most common that you'll see when we get into discussing the UV-vis filters. You also have to consider in your optical instruments how you're holding your sample, how your sample is being contained. I would say cuvettes are the most common. You should think about those materials. Plastic will absorb in the UV, while glass will be more transparent and be more robust, but they're more expensive, while plastic cuvettes are cheaper. And then over here, I have, if you have like a solid sample, in the infrared, you can use salt plates where you dry your sample, and these plates are made of like calcium fluoride, and that doesn't interact with the infrared region. So you really have to think about, with the sample holders, how do they interact with the light itself? You don't want it to interfere. And also, do you really want to measure your sample in a solid state or a liquid state, or rarely in biophysics do we care about the gas state? And then for the detectors, these are all mostly in spectroscopy photodetectors, like I mentioned, that we're trying to go from photons to electrons. And here, older detectors include photomultiplier tubes. Where you have a photosensitive cathode, and then photons hit that, it produces electrons, and there's like a downstream of producing more electrons in the surrounding material, and you can get something like 10 to the 4th to 10 to the 7th gain. So that's how many electrons you get for one photon. Also, there's many photoconducting semiconductors. And these include like silicon photodiodes, silicon diodes, where here you would have like a PN junction. And when a photon hits, you'll have an electron and a hole split. And based on that, you can then detect that electron. And then also charge coupled device detectors as well have a very similar setup, with a potential difference where you have p-doped silicon and a metal. And again, that photon will create an electron hole pair, and you can detect those. So not going into a lot of detail with these detectors, I've uploaded on Canvas a whole book chapter that goes into all the details of these, but realize these work for different ranges. And CCDs are very beneficial for low light levels. And then finally, these detectors have to be connected to a readout. So after those electrons are produced, you have to do the analog to digital conversion. So you can read those electrons on a digitized computer with your spectroscopic instrument. And I've discussed with a lot of these, mentioning like you have to make sure it works in a certain range of the electromagnetic spectrum you're working with. So I've uploaded these tables onto Canvas, and you can see, let's focus up here on this portion of the table. So this is our different sources. You can see some of the ones I mentioned, like the xenon lab works in the UV, a very large range from the UV to the near IR. While if you just have a deuterium lamp, you have a more limited range, the tungsten lamp as well. So if you want to be doing a full spectral measurement from the far UV to the near IR, you're going to have to switch between multiple lamps, and many commercial spectroscopic instruments use that. Here, this line for the lasers represents the type of lasers that you can purchase, but they're going to be at one discrete, one laser is going to be at one specific wavelength. So when you're using an instrument, you should look at, in the manual, of course, it will tell you what wavelengths it works for, but looking at what lamps it's using in those different ranges, or what detectors, if it's using multiple detectors of, let's say you want to measure something in the mid IR all the way to the visible, might switch between detectors as well, and that can have different calibrations and different sensitivities in those ranges. And then again, here's the different gratings, interference filters, the dye-doped, traditional filters, and the different sample cells here. So you can see, like, if you're using a cuvette that's made out of silicate glass, it's not going to work well in the UV, versus if you use fused silica or quartz, so that will be a more expensive cuvette than the typical silicate glass. So these are references that you'll be using in the problem set to note, like, what components that you would use for a measurement. Any questions regarding different components for the optical instruments? Okay. So now let's get into more details specifically with UV-vis absorption spectroscopy. So we're going to be focusing on absorption, but that's one way that light can interact with matter. Since you guys have taken E&M, what are some of the other ways that light and matter can interact? You guys want to throw that in the chat or speak up?
Scattering.
Yeah. Scattering. You think of, I don't know, sorry, the chat's on the side. Okay, there we go. Transmission, we have, which we'll define, is related to absorption. What about if you're using mirrors or different angles? Yeah, reflection, diffraction. We have absorption up there. We're going to get into next week with fluorescence and phosphorescence. There's also refraction. So there's lots of different ways. I would say some have more to do with optics, like refraction and reflection. Some have more to do with, like, the molecular or these quantum transitions as well, and also emission as well. But we're just going to be focusing on that clearly drawn circle, absorption, and I guess transmission with that as well. So if we're going to define absorption, what that means is light is exciting some type of matter to a more energetic state. So it's going from ground, we're probing the ground, to the excited state. These other types of interactions, like fluorescence, probes the excited state to the ground state. But here, absorption is just going from ground to excited. So I've shown this for atomic and molecular sketches out here. where this is our energy level diagram, where higher up on this is higher energy. So we have our ground state here and our excited state here. And I think several of you guys mentioned, I think in the UV and the visible groups, the hydrogen spectrum. So the hydrogen atom spectrum, I'm sure you guys have measured in a lab where you have these nice bands versus wavelength. So with an atomic sample like hydrogen, you'll see very discrete single peaks in your absorption spectrum because you're only probing the electronic energy levels within that atom. But as you have a molecule, you can have additional vibrational and rotational states. So I've shown here in the ground state, that's your electronic state. Then you have vibrations within that ground electronic state. You have vibrational energy levels. And even lower energies are then those rotations. So I've tried to sketch those out there. So when you guys are thinking about those different regions of the electromagnetic spectrum, when I'm asking you those questions about the quantum transitions that you probe, you can see that, okay, electronic, that's UV, and visible, the vibration is slightly less energy, so that's going to be the IR, the next lower, and then microwave is in between probing these smaller energy gaps here. So that can help visualizing. But because there's these additional energy levels, you don't have discrete, well, you have quantized energy levels here, but within the single ground electronic state, you can excite to different vibrational and rotational energy levels here. That causes your absorption to broaden out, or you have a band spectrum as opposed to these discrete peaks with the atomic, if you're measuring an atomic sample versus a molecular sample there. And key to UV-vis and how it's used most often is calculating concentrations. So I'm going to go through and you quantify those concentration with Beer's law. And I want to go through the derivation for Beer's law. So with a typical absorption experiment, you're using your source that has a certain incoming or incident intensity of light. So I'm using I here to indicate intensity, the not meaning that's going to be the starting value. It then passes through your sample. This is going to be some solution in a cuvette. A cuvette has a certain path length, or that we'll call B. And as that light interacts with different slivers of the solution, we're going to call that dx. And then you detect the intensity of light that emerges after interacting with that sample. So related to absorption is the transmittance. And we're going to define transmittance as T, and that's going to be our detected intensity of light over our incident intensity of light. And if we want to understand how the amount of light changes through a certain slice of solution here, we want to understand how I changes with the sliver of solution dx. We're going to say that the change in the intensity is going to be proportional to that starting intensity by some constant. And that constant we're going to define as beta. And it's also going to be related to your starting concentration or your starting intensity, the concentration of the molecules in solution you're detecting, and how big that path length is, dx. So if we want to understand the entire change for the entire path length, we're going to integrate over this. So if we want to understand, okay, let's see if there's a chat. Yeah, quick question, Julie?
Yeah, sorry. So to use Beer's law, are we assuming that the solution is of uniform concentration and composition?
Yes.
Okay.
That's a good question. So yeah, we're assuming that it's same throughout. So to integrate over this, if we want to understand the change in the intensity di, we're going to integrate from the starting intensity to the final intensity I, and that's going to be equal to integrating over the path length from 0 to B. for this expression, negative beta I c dx. So we're going to move the negative I to the other side of this integral. So all the I terms are on the same side. So if we then evaluate this integral, what we're going to get is the negative natural log of I evaluated from I naught to the intensity minus, and then the right-hand term is going to be beta times the concentration times x evaluated from 0 to b. So if we plug those values in, what we're going to get, negative log of I minus the negative log of I naught equals beta c times b minus 0. If we reorganize these natural logs over here using some of the properties of logs, we'll get the natural log of I naught over I is equal to beta c times the path length. So this beta here is just some arbitrary proportionality constant. So we're actually going to change the natural log to log base 10. And then we'll just change our proportionality constant to switch between the natural log and log base 10. So that new constant we're going to call eta times c times b. So we can also flip the order, again, using properties of logs. If we take the negative log of this, it's just going to flip what's inside the log term. And this will give us that the negative log, this is the same as what we define as the transmittance, is equal to eta CB, which is then what we call absorbance. So this is Beer's law, where your absorbance depends on your path length, your concentration, and then this constant here, eta, is what we call molar absorptivity. And this has units of 1 over centimeters times molarity is the typical units. And it is a value that's a property of whatever you're measuring, whatever concentration, whatever molecule you're measuring. And it typically ranges from 0 to 10 to the 5th inverse mole centimeters. And this is dependent on wavelength, it's dependent on the solvent, and it's dependent on the analyte. So this is something, if you're doing an absorption measurement, that you need to look up and you need to know about your sample. But I'd say that UV-vis spectroscopy is typically used to look at concentrations in relation and using this Beer's law. So some applications of it being used in biophysics, I would say, can be used as is with amino acids, proteins, and nucleic acids, because it is probing, again, we're looking at electronic transitions. And you can see that these structures for amino acids or the nucleic acids, that it has that conjugated structure. So conjugated molecules have their pi electrons, those transitions in the pi electrons. So with these conjugated molecules, you kind of have a cloud of electrons over the entire structure here. And it's a fun exercise if you guys have taken quantum chemistry or modern physics, that if you look at the length scale of these molecules and you do particle in a box calculations, the energies for particle in a box of about, let's say, like 500 picometers corresponds to the UV-vis range. So I would say for amino acids and proteins, it's used to determine concentrations based on tryptophan, tyrosine, and cysteine that I have shown here. That because these have the conjugated structures, their electronic transitions correspond to wavelengths of around 280 nanometers. And this is in the problem set, problem number 6, that you'll work through figuring out the concentration of a protein by looking at the absorption of these amino acids. You can also use the visible region if you have fluorescent proteins. So these are, we'll talk about this when we get into fluorescence, that they have these beta barrel structures and they absorb in the visible range. If you guys have seen green fluorescent protein, let's see what the question is. Is histidine technically not resonant? I'd have to look at the specific wavelengths for histidine. I think because of the number of amino acid groups, or the number of nitrogens and amines there, that histidine is actually shifted a bit bluer. So you can see like with the nucleosides here, I think for histidine, it shifted more around 250 or so nanometers, but I'll get those specific numbers. So in the visible region, if you use these specific tags or conjugate or make a protein that has something that absorbs in the visible region, you can also do measurements there. For nucleic acids, I have the nucleobases shown here again, those pi conjugated systems, that they typically absorb around 260 nanometers. You'll also see that cells are measured by UV-vis, but they're used for scattering. So since cells are around, let's say if you're working with bacteria cells or yeast, like 5 microns or so, they scatter light, and typically to look at their concentrations or their growth over time, the scattering is studied at 600 nanometer for cells. And then finally, for other biomolecules, colorimetric assays are used. So this would be the fact that, like, let's say you're trying to detect a lipid that has this long hydrocarbon chain or so. This is going to be way in the far UV or further to be able to detect any absorption there. Or let's say with carbohydrates or any other type of biomolecule you may be interested in, where you want to look at the production of ATP that absorbs again in the far UV that can't be detected on a UV-vis instrument that typically only goes to the near UV, maybe mid UV. So with this, you have some type of color small molecule that reacts with your biomolecule of interest. So here I have an example of like a 96 well plate where these colors represent, it's a small molecule reporter that turns pink if there's hydrogen peroxide, if you're trying to detect how much hydrogen peroxide your cells are producing, and it remains blue if that hydrogen peroxide is not being produced. So there's many different types of assays or colorimetric reporters based on what you want to detect. Small metal ions, if you want to detect ATP itself. So I'd say that's a very common way that UV-vis is used in biophysics. I do want to talk about a little bit about like practical use of the UV-vis spectrometer. I know that this isn't a lab course, but I'd like you guys to understand that there are, if you do go in the lab, what you might see, how you might use this instrument, what you should consider. One thing is with the instrumentation, I would say UV-vis is like the most common type of instrumentation you'll run into from different vendors. So you'll see them made by many companies, but Carey and Agilent, Shimadzu, Beckman Coulter, any Thermo Fisher, BWR, they'll all be making their own type of UV-vis. It can range from being on the bench to being handheld. There's different sizes depending on like how precise, how accurate, what we discussed in lecture 2, how much detail you need. If you're just checking a single wavelength at 600 nanometers for cells, it might be the size of a large cell phone. In footprint, if you're trying to do very thorough, very sensitive measurements, it can take up an entire desk type area. It's worth noting that fancier UV-vises will have double beams, which I show here, that you can place a sample and a reference at the same time and detect the signal from both of those. So if you have a time-dependent process or a more sensitive sample, having a measurement of the reference at the same time as the sample can reduce the amount of noise if there's environmental noise. But those are going to be more expensive instruments. And also, if we're going to talk about BioBox, and just like those assays that in more biofocus lab or people who are doing more assays, you might have UV-vises that can work with these 96 well plates. So it's able to scan through robotically, taking measurements at every single well. If you're doing drug screening and you're testing hundreds of different drug candidates, you want to be able to take measurements and read this out very quickly instead of doing it all by hand. So those are different arrangements of UV-vises that you may see. When you're taking an actual measurement, it's important to work within the range of the instrument. So working in the middle of the range, 0.4 to 0.9 absorbance is good practice. If you're close to 0 or close to 1, remembering that this is a log-based measurement, you're not going to be detecting very many photons. If you're absorbing all the light, you're going to be working at the limit of your detector, or if you're at 0, detecting a small difference is going to be challenging. You also have to think about, with your sample, that you should be working at a concentration range where there's no aggregation or scattering, and you want to be working at equilibrium. That if you're working at very high concentrations and you have aggregation, you're not going to get an accurate concentration. If you're having a chemical process take place during your measurement, it's not going to be an accurate concentration. Also with this sample, thinking about the cuvette you're using, you want to make sure it's clean. You want to avoid systematic error. So if you use the cuvette in the same direction every time, the same cuvette, you're not going to have any strange results from, let's say, one cuvette has a path length that differs by a couple, like 50 microns, 100 microns. If you use the same cuvette over and over again and also facing the same direction, you're going to avoid those type of errors. And also, if you're doing a calibration curve, you always want to work from a low concentration to a high concentration. So you don't have to like clean thoroughly every single time. If you're working from high to low, you're probably going to get inaccurate measurements. So these are some tips and tricks to avoid those systematic errors. And another thing to point out is to understand the settings on your UV-vis. So you get a spectrum and you take a point and you're like, oh, I'm getting a value at a single wavelength. It's exactly this. But realize that you're using lamps with the UV-vis. And with your grading or your wavelength selector, it's not a single wavelength, it's going to be a band of wavelengths. So how how small is that band and what is your value there? Realize that this isn't a monochromatic measurement. So any questions with the UV-vis at all? Okay. And I had one more, let's see. I guess we have 5 minutes. So instead of going in the breakout groups, I do have this question here, that if you're taking a UV measurement, let's say we're taking a measurement of just a solution of tryptophan, that amino acid, and you're measuring it in a 5 millimeter cuvette, and this UV-vis has a deuterium and tungsten lamp. You take your measurement and you get this dashed curve here, where you see this jump, you see this increase as you go into the UV. Do you guys have any ideas why your spectrum would look like this? Both the jump and that increase. You guys want to add in the chat if you want to think about it. Any ideas? So there's some clues in the, okay, let's see what's in the chat. So aggregation, increase in absorption. So if you're looking at a high concentration, we might expect some aggregation. There's some clues in the question here. So
from the plastic cuvette interacting with the at lower wavelengths.
Yeah, so what Bill said, and also Catherine says, difference in calibration of the lamp. So yeah, and Dushani mentioned switching of lamps. So the plastic cuvette would be the reason that you're having this high absorption in after you pass 300 nanometers or so. Or I guess after like 280 or so nanometers. So the jump here, this discrete jump would be you're switching between your tungsten lamp that works in the visible and the deuterium lamp that works in the UV. So that's why the jump is there. And then the high absorption after 270 or so is because the plastic absorbs there. So the way you would correct for this would be measuring in your quartz cuvette and also calibrating, like Catherine says, calibrating the lamps and doing calibration measurement at the start. And then I'll leave this question for you guys if you want to look through it. It's in the notes. I don't know if I put the answer in the notes, but if you want to check what your answer is, this is another walkthrough of calculating concentration using Beer's law, but is also in the problem set as well. So let you guys know there's also, what's going on here? I did put some references on Canvas that go into a lot of detail. The Skoo Collar Crouch, I actually uploaded the whole book, but chapters 6 and 7 would be relevant to this lecture. And then chapter 3 of the Biophysical Tools book talks a little bit about spectroscopic information, but it's going to go into a lot of detail of fluorescence microscopy, which we'll talk about next week. So any questions at all? Okay. Thanks for the lecture. Hopefully the breakout groups went okay. Any feedback with that, feel free to upload to Canvas or email to me. So have a good rest of your day, guys.